In the evolving landscape of enhanced oil recovery, the gap between laboratory success and field application remains a critical challenge. Today, our group will be critiquing the paper titled Microbial Community Distribution in Low Permeability Reservoir and their Positive Impact on Enhanced Oil Recovery by Pang et al. Understanding how microbial enhanced oil recovery works can help improve efficiency, reduce costs, and provide a more environmentally friendly solution for oil production. Our goal today isn't just to summarize the findings, but to rigorously evaluate the methodology, the validity of the data, and the practical implications for the industry. I'm Haima and I will handle the introduction, followed by Elena with the article summary. Jocelyn will lead our co-critic, while Franz will synthesize our key takeaways and Hakimi will close with future research directions.
Let me now walk you through the summary of this article. The primary of the study was to investigate the distribution patterns of microbial communities in low permeability reservoir and understand how these microorganisms contribute to enhanced oil recovery. Specifically, the researcher focused on the S169 block, a typical low permeability reservoir in China. The authors hypothesized that microbial communities, despite the harsh reservoir conditions of low permeability, high temperature, and high salinity, they can actively grow and produce metabolic byproducts that reduce oil-water interfacial tension, improve crude oil fluidity, and ultimately increase the oil recovery efficiency. To test the hypothesis, the researcher utilized an experimental approach combining sampling with laboratories analysis. Two categories of water samples were collected from the S169 block, which is the enrichment pond waters and wellhead produced water samples. These were obtained in two separate batches to capture the microbial changes over time, which is important because the microbial populations are dynamic and respond to environmental and operational conditions. So the methodology consists of four key stages, which is the first one is microbial concentration measurement using the plate count method to quantify the growth. Second is metabolic analysis that using the gas chromatography, GC and GCMS, gas chromatography, mass spectrometry to detect organic acid, lipids, solvents, and gases produced by the microbes. The third one is the functional gene quantification using the quanti-PCR or qPCR, targeting markers such as cysteine-S rRNA for bacteria, ARCH for archaea, and MCRA for methanogens. And lastly is the high throughput sequencing to evaluate microbial diversity, community composition, and structural changes across the sample. So the result indicated a significant increase in microbial concentration across all enrichment ponds, with growth rates ranging from approximately 6% to 16% between the two sampling batches. More importantly, metabolic revealed a substantial increase in lipid concentration, up to 287% in one pond, suggesting intensified microbial metabolism and biosurfactant production. Functional gene analysis demonstrated a temporal variation in bacterial and archaeal abundance, while sequencing results showed that microbial communities were highly heterogeneous and strongly influenced by geological factors such as porosity and permeability. Essentially, the authors concluded that microbial activity contributes positively to enhanced oil recovery by producing metabolites that improve crude oil displacement and mobility, and that the distribution of these microbes is closely tied to the physical characteristic of the reservoir. While these results appear promising on the surface, a deeper look into the experimental design and data integration reveals several areas for discussion, and that's exactly what my colleague will now address.
The technical framework of our article is robust, utilizing a high-throughput 16S or RNA sequencing and qPCR for quantifying gene abundance, paired with GCMS for metabolite profiling, successfully characterizing the shift in microbial community structure and production of biosurfactants and gases. However, the research exhibits vulnerabilities, such as the absence of negative control, failing to isolate the treatment effect from natural reservoir flux or abiotic geological factors. The sample method also lacks transparency as the transition from 24 wells to 19 is not properly justified by power analysis nor selection criteria, introducing a potential selection bias and obscuring validity. Now, regarding biological scope, the reliance on the CQM3 limits our understanding of microbial synergy, since the study is confined to only one block. The findings are constrained by specific variables such as permeability, while the biochemical mechanisms such as interfacial tension reduction are scientifically sound. The delta in recovery rate as well cannot be extrapolated to high permeability nor high salinity carbonate reservoirs without further multi-site variability validation. In the replicability realm, it's where its higher applicability lies. As the documentation of PCR thermal profiles and instrument specifications are exhausted and well documented. The use of two sampling batches are also added reliability, and this topic is also very important in petroleum industry research, contributing to more profitable recovery options.
So the study shows a strong and scientifically grounded examination of the impact of microbial communities on reservoir performance. So by establishing a clear integration of biology and also geology, this microbial synergy drives oil recovery through production of metabolites, which significantly enhance the crude oil mobility. So although the stats comes out with significant advantage, it also has some drawbacks, which are the absence of control group, the lack of exponential reduction wells, as well as the limited geographical scopes, which suggests that the funds might not be reliable because it focuses only on significant geographical region.
Based on our critique, future research should focus on addressing the main limitations of the study, especially in terms of long-term sustainability and wider field applicability. First, future studies should involve multiple reservoir sites with different geological conditions. This is important because testing the method is only limited settings may not fully show whether the findings are consistent and reliable across different reservoirs. Second, researchers should include proper controls group and longitudinal monitoring. This would help evaluate the long-term stability of microbial communities and also determine whether the recovery performance can be sustained over time, not just in short time. In addition, future research should further investigate the specific synergistic roles of microbial metabolites, especially biosurfactants, organic acids, and biogas. Deeper understanding of how these metabolites work together would strengthen the mechanistic explanation of microbial enhanced oil recovery or MEO. Another important direction is the integration of reservoir simulation models with real-time genomic monitoring. This could improve predictive accuracy and help optimize MEO strategies more effectively for industrial application. From an interdisciplinary perspective, this area of research also has strong potential to align with the United Nations Sustainable Development Goals or SDG, particularly in promoting more efficient resource use and reducing environmental impact in oil recovery operations. In conclusion, while the authors have provided a valuable starting point, the journey toward a fully optimized and widely applicable MEO process still requires more robust validation. Thank you for listening. That's all from us.